Good evening. Thank you all for joining us tonight on the first event in our RiverWatch Winter Speaker Series. For those of you who are not familiar with the Illinois RiverWatch Network, RiverWatch is a citizen science organization that trains volunteers to monitor water quality in their own communities. The mission of RiverWatch is to safeguard the future of Illinois rivers and streams through stewardship, education, and sound science. Before we get started, I would like to ask you to keep your microphone muted during the presentation to limit any distractions. We will have a Q&A session after the presentation, so if you have a question along the way, you can either put it in the chat box to be answered at the end, or you can save your question and ask it aloud. And with that, I would like to welcome Dr. Tom Anderson. Stop sharing so he can get his set up. Tom is an assistant professor of biological sciences at Southern Illinois University, Edwardsville. His research is focused on factors that help generate structure in aquatic ecosystems, including competition among species and predator-prey interactions. He works most frequently with amphibians and aquatic invertebrates, and tonight he will be talking with us about amphibian phenology and climate change. Tom, I'll let you take over.
Great, thanks Danielle. I'm happy to chat with everyone about some of the work that I've been doing and some of the things that interest me in terms of my sort of scientific pursuits. So first of all, can everybody hear me okay? Is my audio doing good? Okay. So what I'm going to be talking about tonight is amphibian phenology and climate change. And so this sort of a roadmap of what I'm going to be talking about. So I'm going to start out just talking generally about pond breeding amphibians, which is most of the organisms that I work with, and then getting into a little bit of sort of general information about phenology, if you're not familiar with that term. And then sort of ending the talk, talking about methods that researchers like myself use to study and quantify phenology for specifically for amphibians, but I'll have some sort of general information as well, just about what folks do to try and understand phenology. And so this is the area of my current research with amphibians, and so it's sort of exciting as we start to transition out of winter here to start to think about, organisms coming out of their hiding places from underground and then starting to emerge. And so, thinking and putting this talk together, started getting me excited again, thinking about the upcoming field season that I'll be partaking in. So first, though, for pond breeding amphibians, so if any of you were at Mark Davis's talk on Monday night from the Illinois Natural History Survey, he gave a very nice overview of all of the amphibians and reptiles of Illinois. And so I'm just going to go through a little bit of background information about pond breeding amphibians and the organisms that I work with. And so I have pictured on here a variety of the different species of frogs that use ponds or wetlands as part of their life cycle. And so these are some of the more common species that you might have seen, in wetlands near your house or in your neighborhoods that you live in. I've seen several of these species in my yard here in Edwardsville. So things like American toads. I frequently hear gray tree frogs calling from my back porch in the summer, and a variety of other critters that many of you have probably seen or heard. And so the organisms that I primarily work with, rather than all of these frog species, though, are salamanders. And so salamanders are creatures that many of you may not have seen. I never saw a salamander until I started my master's program down at Murray State University down in Kentucky. And so I made it through into my 20s before having ever seen a salamander in my life. And part of the reason that you hardly ever see salamanders is that they spend the majority of their life underground for up to 9 months out of the year. So they're just not very visible, and they aren't making noises like many frogs are. And so they're just much harder to observe. And so shown here, I have a variety of different species of salamanders that are all in the same family, collectively referred to as the mole salamanders. And so the species and the 4 on the left here, and then the top one up here on the right, are all species that are found here in Illinois. The bottom right species here is a species that's only found down in the Ozarks and Ouachita Mountains of Missouri, Oklahoma, and Arkansas. And these are sort of just a representative set of species that you would find sort of in the general region around us that are usually fairly common. So in my part of Illinois where I live, so the smallmouth salamander down here and then the tiger salamander up here are the two main species that I frequently encounter where I live. And so these species, because they're relatively secretive, my former PhD advisor used to refer to these organisms as clandestine creatures of the night, as sort of a collective phrase that he used to describe them because they're very sort of secretive in nature and just not observed very often, again, because they spend much of their life underground. So both frogs and salamanders, for the most part, for the ones that live in ponds at least, undergo what's called a complex life cycle. And so what this means is that their life cycle is split across both a terrestrial and an aquatic, or their life cycle is split across terrestrial and aquatic habitats. And so adults are primarily terrestrial, living somewhere in upland habitats surrounding wetlands or ponds, usually somewhere in the neighborhood of within 200 to 300 meters from a given pond that they might be using. And so during certain times of the year, the adults will emerge from underground and undergo mass migrations to lay eggs in aquatic habitats. Those eggs develop in the water, they hatch out and turn into either larvae, which is what the term is referred to for salamanders, or tadpoles, the term for frogs. And those larvae or tadpoles spend anywhere from a couple weeks up to many months hanging out in the water, growing and developing. And then after, at some point when they reach sort of a minimum size threshold, they undergo metamorphosis back to the terrestrial environment, where they then spend anywhere from 1 to 3 years as a terrestrial juvenile before they then reach sexual maturation and become a breeding adult. And so this complex life cycle revolving back and forth between the terrestrial and the aquatic habitats, depending on which life stage it is. So just to show some examples of what this complex life cycle is for a couple species. So again, this is the ring salamander that's found over in the Ozarks and Ouachita Mountains. So these are the terrestrial adults. They lay these aquatic eggs. Each of these individual little black dots is an individual embryo. And so they'll lay anywhere from 100 to 300 eggs for each female will lay. Those eggs develop after a couple of weeks and turn into these larval salamanders that have these big frilly gills that they use to take in oxygen. So many of you probably have heard of axolotls. This is what a lot of larval salamanders look like. Though, unlike axolotls, these eventually will transform out of the aquatic environment into these terrestrial juveniles, where they'll spend, again, several years underground before reemerging as a terrestrial adult. And so just another example, same sort of thing. So for slightly different egg mass for spotted salamanders, a common species in the lower third or so of the state of Illinois. Again, aquatic larvae, terrestrial juveniles, and adults. All right, so I'm pointing out all of these different life stages in this complex life cycle because it relates to the concept of phenology. And so phenology is this idea of the timing of seasonal recurring biological events, or some sort of biological process that has some sort of timing component to it. And so for amphibians, some of these phenological events are things like the timing of these breeding migrations that the adults undergo when they migrate back to ponds to lay their eggs. Other phenological events could be things like the timing of eggs hatching, or the timing of when those larvae or tadpoles undergo metamorphosis back to the terrestrial environment. And so any of these sorts of biological events are referred to as the sort of different phenological processes that amphibians undergo. Though phenology is not a term that's uniquely applied to amphibians. Phenology can be referred to any organism that has some sort of event like or life cycle like this. So it's often used in plant research. So the timing of when buds burst, or when flowers or leaves emerge, and those sorts of things. The timing of bird migrations, or the timing of emergence from hibernation, like a lot of mammals do. All of those things are different phenological events. These are the ones here I've listed here, just are specific to amphibians. So in terms of these events, breeding migrations are by far the best studied phenological process that amphibians undergo. And part of the reason is that, especially for salamanders, this is one of the main times when they're actually visible to people when we're out looking for them. So if you've ever driven out on a rainy night in March around here, you'd often potentially come across seeing adult salamanders crossing the road. And so this would be when they're undergoing this migration back to wetlands to breed. And so just because they're the most visible during these stages, as well as frogs, that's when they're all calling. So breeding time periods are by far the best studied events in terms of phenology. And so we know a lot about what causes amphibians to emerge from their underground hibernation sites or underground burrows or wherever they're hanging out. We know what factors drive them to initiate these movements back to wetlands to breed. And so temperature and precipitation are the main things that are the cues that amphibians use to emerge out of underground. And so most amphibians have fairly skin that needs to stay fairly moist. And so that precipitation allows them to be able to move above ground without having a lot of risk of them drying up too much. And then as well as having suitable temperatures, if it's too cold or too hot, then they're unlikely to be up moving around just because it's too costly to them in terms of their physiology. And so just as an example of the types of data that folks look at for understanding how these cues initiate movements, some data that I was able to work on with some folks that I did my PhD with, we looked at the timing of movements of adult ring salamanders at some ponds over in Missouri. And we looked at the number of adults captured here on the y-axis as it relates to how many days had elapsed since it last rained. And as you might expect, the longer it had been since it had rained, we caught fewer and fewer adults. And so those main capture events were really tied to days where we had a lot of rainfall happening. So the highest captures were on the days that it rained or were the day after that it rained. And so these sorts of things are the main cues that are causing this phenological process of breeding migrations to occur. So one of the things that's of concerning to many scientists, again, for amphibians and a variety of other organisms, is that phenology is changing for species worldwide as the climate shifts. And so as temperatures in the spring come earlier and earlier, so as spring arrives earlier and earlier, these many amphibian species are also shifting when they undergo these breeding migrations to wetlands. And so many taxa have shown earlier shifts to breeding as that climate begins to warm. But it's interesting that not all species are changing in similar way or at similar rates. So for example, this is a table from a paper that was written in 2011 by Todd et al. And what I want to highlight here is that each of these different rows is the scientific name of a different amphibian species. This study happened to be done down in South Carolina, and they looked at the estimated calendar arrival date and from the beginning of the study period to the end of the study period. So spanning, almost 25 years for a lot of these organisms. And what they found was that over this time period, each of these different taxa either showed a substantial shift in the estimated change in the days that they underwent their breeding migrations. And so, for example, in this first row, this species is the four-toed salamander, was breeding 76 days earlier than it used to at the beginning of this study period. The next species, the marbled salamander, 15 days earlier. In other cases, I said that backwards. These are later, 76 days and 15 days later than they used to breed. Here for the tiger salamander, it's breeding 56 days earlier and chorus frog 60 days earlier. But then all of these values that say NS here mean non-significant, meaning that species phenology didn't change. And this is interesting because all of these organisms, this is from one wetland. So some organisms at the exact same pond were shifting by upwards of almost 2 months, whereas others were not shifting at all. And so there is a really strong species-specific response to climate change that might explain why, that is related to how well or how sensitive they are to the climate around them. That some species are really sensitive, whereas others are not. So the two unknowns that I want to cover then for the rest of my talk, or that are two unknowns generally in phenology, are, where are these phenological shifts happening? So what populations are undergoing these phenological shifts? And then what type of shift are they undergoing? And so that data that I just showed on that last slide in that table was for the median date of breeding. But there are also other types of shifts going on. So it could be the date that the first individual arrives or the last individual arrives, or some other sort of mathematical quantity that you could calculate for other types of shifts, as well as I already mentioned, who is shifting their phenology, what organisms are shifting and who is not. But then the bigger question that's sort of greater importance is, what does these shifts mean for these organisms? Is it a problem if these salamanders and frogs are shifting to breeding earlier or breeding later? How does it affect individual species? How does that affect entire amphibian communities, as well as the overall ecosystems that they live in? And so to go about tackling both of these questions, we need good ways to quantify or study phenology. And as I mentioned, because some of these amphibians are relatively hard to study for most of the year, you're sort of confined to certain windows of time during the season that you can actually collect data on these organisms. So for example, when they're out crossing roads like this adult ring salamander is. If you contrast this with studying plants and you wanted to know whether or not plants are changing the timing of when they put out their leaves, a tree is generally always going to be in the same spot from where you last saw it. And so you can just go back out to that tree and see what it's doing. These organisms, it's a little bit more tricky to do that. And so what I'm going to talk about first is some of the different methods that I've used or others have used to try and understand how phenology, or how we understand what these organisms are doing in terms of their phenology. And then I'll finish up talking more about the consequences of phenological shifts to a variety of organisms. So first, for some of these methods that we use to study phenology or monitor these phenological events from occurring. And so one method that's been effective that the study that I showed for the that previous table is something called a drift fence. And so for a lot of wetlands, you can install a fence that's buried, 10 centimeters underground surrounding a wetland. And then along this fence, you can install these pitfall traps that as amphibians undergo their breeding migrations into a wetland like this one, they get intercepted by the fence and then walk along it until they fall into one of these pitfall traps that you can then go out and collect them and look at the sexual status of them. You can measure their body size and then release them on the other side of the fence to continue on their way back into the pond. And so it's a nice way to effectively survey a population, both in terms of getting the numbers of individuals of different species, but then also you have a really good and thorough record of the timing of when those events happen, because you're out there checking these every single day to see when individuals are coming in and going out. You know exactly to the day when all of these events are happening. And so just to show you some examples of the kinds of data that you can generate with a drift fence. So here on this graph, I'm showing data for two different species. So ring salamanders, again, here in the gold box, and then spotted salamanders here in the gray box. And so this was data from a population over in Missouri that had one of these drift fences installed around it across 4 years. So 2004, 2005, 6, and 7. And they had this drift fence in place and they monitored it year round. And they looked at the number of adults they captured, which is what the y-axis here is showing, across all of the different days of the year. And so each of these bars then extends up to the height of the number of individuals that were captured. These are binned, I think, into 2 weeks in terms of total captures, or a week between each of these different bars. So, or a little over 750 individuals captured over about a week period. And so what you can take away from this information using a drift fence is that you can see variation in the timing of breeding, both across species as well as across years. And so, for example, in spotted salamanders, we see that in 2004, they had this really big spike and then a very few smaller peaks in terms of numbers of individuals captured. Whereas the next year, they had a much flatter distribution of captures that were much less in terms of one peak being really large, but were much more sort of consistent in terms of their height, around 250 individuals captured. Similar patterns for ring salamanders, where you can see they have somewhat similar shapes, but there is some variation among years. And so this is a really good way to document what phenological patterns are like now, so that way we can better understand what they might be doing in the future. And these drift fences are really nice in that you can leave these up for a long time and they're relatively low maintenance. And so you can generate really long studies if you're able to research the same location for a long time, to be able to track what that population is doing over time. So where researchers have had drift fences set up for upwards of 40 years at the same pond to be able to track changes in numbers of individuals, as well as things like the timing of breeding. So the drawback of doing this kind of work is that it does require you to go out there every day to check what's going on at the fence and to make sure that you've captured individuals and then release them right away. So one method that's been sort of an emerging technology that we, myself and some collaborators used, was to start to implement using trail cameras to try and photograph these migration events, as opposed to having pitfalls buried in the ground that would then have to require you to go out and pick up animals out of the pitfalls. And so what we did here was that we set up a similar sort of fence around a pond. And so the pond would be off the bottom left corner of this screen. We left an opening in the fence that we put this cork board over the top of to create a nice contrast in the background, and then mounted a typical wildlife trail camera pointing downward at the ground. And then it would take a picture every minute all night long from about dusk to dawn. So, once a minute, so, several hundred photographs every single night. And we had hoped to capture pictures then of migrating salamanders coming into these wetlands as well as leaving. And thankfully, this actually worked to some degree. So this is a picture of a ring salamander that we got a photograph of as it entered through the opening in this fence into the wetland. And so this was a nice remote way of monitoring breeding phenology of salamanders that didn't require us to be out there. And we'd only have to go pull the cameras out of, or the memory card out of these cameras once a week to download the pictures. And so it was a much less intrusive way of doing this, as well as having then a photo record of all of these animals. And we could generate similar sorts of data for the breeding, just like we had on the previous graph. And so here again, we have the total number of adults. This is from a pond in southern Missouri down near Fort Leonard Wood, where we had this camera set up. And so this was from the year 2017. And so again, we have day of the year on the x-axis. And then for two different species, ring salamanders and marbled salamanders, we can see the number of individuals that were captured coming in on each of these days over about a 2-month period. And as you might expect, a lot of these movements were tied to rainfall events that occurred in this area. So where these big spikes occurred were on the same days that rainfall events occurred. And so this was an exciting way to try and start to do some things that required a little bit less person effort to be going out there every day. However, we did generate, over the course of 3 years of having these cameras out there, we generated over 600,000 photographs that we needed to sort through and find pictures of salamanders, most of which did not have them. Because it was taking a picture every minute, there's a lot of empty photographs that we got that we had to sort through to try and find this information. And so these trail cameras, though, because they're such a great tool, a lot of folks have been working on modifying this to where instead of it requiring you to take a picture every minute, you can actually set up devices that would essentially be a laser beam going across the opening of this, that as animals walk across it, they trip the laser beam and trigger the camera to fire, which then it would reduce a lot of those empty images that you have to sort through. So I think this is sort of a great way moving forward to try and do some of this work. So moving out of just salamanders now, some other ways that folks are really using to be able to monitor the timing of breeding in vocal animals like frogs is putting out a variety of acoustic monitoring devices that are automated to turn on and off at specified time periods to be able to record whatever is going on around them. And so I have some pictures just of a couple examples of these different monitoring devices. So this one on the left here is a relatively recently developed device that costs around $80 or so that you can buy, and then you can set it to record. The ones I have set to record for 2 minutes every hour from all night long around a variety of ponds around the SIUE campus. And so that way I can keep track of the timing of frogs when they start calling in the spring, as well as the duration of period over which they call. Same sort of device here on the left. This one's a little bit more expensive, more in the neighborhood of around $200 to $300. But these are, really useful tools, again, for sort of remotely monitoring a variety of wetlands. And so something that I failed to mention back when I talked about drift fences is that usually when you set up a drift fence, you're constrained usually to doing that at only one pond. It's simply too much work unless you have a really, really large team of folks working on it to do those at many locations. Things like these are much easier to put one of these at a variety of different wetlands to get a really large spatial sample of what frogs are potentially doing in terms of the timing of their breeding. You could put these, 20 of these out at different wetlands across a large area and record what's going on. But again, obviously salamanders are not making a lot of vocal calls, and so this would not work for monitoring what they're doing. And so then the final thing, and this is sort of a pseudo plug for a program that I recently became aware of, but a lot of community science projects are becoming really effective tools at monitoring the phenology of organisms, amphibians and otherwise. And so using apps like iNaturalist or other similar sorts of programs are really effective ways of documenting what organisms are doing at different times of the year. And all of this data is then available for researchers to go in and look at what different users have submitted in terms of finding frogs calling or adult salamanders crossing the road, and being able to then pull information out of that to try and make scientific conclusions about what's going on at a much larger scale than any single researcher would be able to do by themselves. And so one program that recently started that I'm excited about being involved in is this program that the Midwest Partners in Amphibians and Reptile Conservation, as well as the program HerpMapper, have put together basically to track phenology of amphibians and reptiles in the Midwest. And so this is through the website HerpMapper, where you can submit these observations that get pulled into this database to try and track phenology of organisms in our area. So if you're interested in this sort of thing and want to participate in phenology type work, then these sorts of programs would be ways to be involved. So these are sorts of ways of studying phenology in the field in terms of monitoring natural populations. But like most scientific studies, there are often limitations that also come along with a lot of those types of that type of work. And so some of the things that are sort of problematic for doing field studies, trying to look at phenology, is that if the goal is to understand whether or not phenology is changing, we often need really long time series. So basically a lot of years of data before we can really say a lot as to whether or not phenology is changing. So the study that I showed the table from was over 25 years. And so, that's a really long time to be studying one spot to be able to say much about it. And again, things like drift fences are limited in how many ponds you could put them at or wetlands you could put them at. And so it has a limited spatial extent, whereas the other things that I mentioned sort of are a little bit less so. And some of these are potentially high cost and effort. So again, having to check a drift fence every day, or potentially putting out a lot of the audio recorders, potentially can add up in cost if you want to put them in a lot of wetlands. And so one thing that amphibians are really useful for is that they are a really good system that's tractable for experimentation to try and understand what might happen for sort of different phenological shifts that might occur. And so one of the ways that I do this is I use what are called outdoor mesocosms to try to understand what's happening in ponds under different scenarios of phenological or shifts in phenology with breeding. And so what these are in this picture are 1,000-liter cattle watering tanks that you can fill up and basically build a pond ecosystem from the ground up, adding in things like leaves or plankton or a variety of other organisms to build a facsimile pond, but you still have control over it to some degree of what goes in. And in particular, being able to put in certain amphibian species or adding them at certain times, so you can still do a reasonably good experiment in terms of having control over it. And in terms of phenology research with amphibians, one thing that's really useful is that you can often alter the timing of egg hatching for many amphibians by altering the water temperature that they're reared under. And so eggs will develop much quicker under warmer water temperatures than colder water temperatures. And so you can use that, that feature of their eggs to simulate different scenarios of breeding or hatching patterns. And so if you put some eggs in warmer water and get them to accelerate development, and then you could track what happens to those subsequent hatchlings in ponds, or you could also delay hatching to see sort of different scenarios of what might occur. So this would get at that question of, do these phenological shifts matter for organisms down the road? And so I'm just going to talk about one experiment that we did with marbled salamanders. And so this is the adult marbled salamander here. And so this species is found more down in the Shawnee National Forest area of Illinois, so the southern part of Illinois, as well as a lot of the eastern United States. They're a little bit different than a lot of pond breeding amphibians in the life cycle I showed before, because they actually lay their eggs underground, or not underground, but on the surface of the ground underneath things like downed trees or underneath leaves. And those eggs remain on a dry basin until a pond then refills, potentially after being dry all summer, would inundate those eggs with water and then allow those eggs to hatch. And so we wanted to understand in this experiment, how does hatching synchrony in marbled salamanders affect things like survival to metamorphosis, or how long it takes them to complete metamorphosis, which are proxies for potentially long-term sort of outcomes for that population. And so by hatching synchrony, what I mean is that we had 3 treatments where we hatched all of these eggs on the exact same day. We had a medium synchrony treatment where we hatched them out, spread out over a 6-week period, but hatching them on 3 days. So basically week 0, week 3, and week 6. And then a low synchrony treatment, meaning that we hatched eggs out each week for 6 weeks. And then, and by hatching them out, what I mean, because these are laid on the ground, we can put them in containers like these little plastic containers here, and then fill these containers with water to induce hatching at certain times to mimic what would happen under a year where all of the eggs hatch simultaneously, versus the low synchrony treatment would be an example where breeding is really drawn out and a pond might fill incrementally. So every 10 centimeters going up and up and up. And so eggs would be inundated at different times, resulting in a really low amount of synchrony of hatching among all of those eggs. And so we raised these marbled salamanders in tanks just like this, and tracked them for over about a 9-month period, which is how long the experiment took in the aquatic environment. And along the way, we started measuring things like larval size throughout the experiment at a variety of time points, and then again, tracked them up through metamorphosis. And so just to show you some of the data from this experiment that we were excited about is the first, I guess, to orient you to this graph. So on the x-axis down here, we have calendar date, not Julian date, that's a typo. Calendar date that we went out and captured larvae in these tanks that we put them in. And so I caught them on 4 different days, January, March, and 2 days in April. And we measured their total length from photographs like the one taken here. So we could put them over top of a ruler, measure them, and then re-release them quickly, photograph them and put them back into their tank really quickly, and then use the computer program to measure how long each of these individuals was at different time points. And so that's what the vertical axis is here, is the total length of larval salamanders. And these are color-coded by our different treatments, our high synchrony treatment, where all individuals were hatched on the same day, versus the medium and low, where they were spread out in their hatching over a period of 6 weeks. And so what this graph shows, or what I want you to take away from this, is as we start here in January, you'll notice that all the blue dots are clustered right around 30 millimeters here, whereas the green and yellow dots are spread out, ranging all the way down here to around 18, all the way up to around 45. And so the variability in total length for our medium and low synchrony treatments was really, really high, whereas when all the individuals hatched on the same day, they were all roughly the same size. And so this is exactly what we would have predicted, is that when you spread out when those eggs hatch, the larvae that result from that are going to be much different sizes, because they've had differing lengths of time to grow up until this point in development. But as we transition from left to right here through these different days, you'll notice that the green, the blue, and the yellow all start to intermix in terms of their spread around here, to where in April, the blue, the yellow, and the green are completely intermixed with each other. There weren't any differences among our treatments in terms of how big these larvae were. So another way to look at this, if we look at, instead of it being total length, if we change that around to a different quantity of variability in total length, now we have each dot on here is the variability for one tank in each of, again, these treatments for the high, medium, and low. So the colors are the same here. We can see that variability in our medium and low synchrony treatments really declined through time, whereas our high synchrony treatment basically remained the same in terms of the variability in body sizes. So larvae were growing, but just they were all about the same size here. Whereas up in these treatments, they started out really different in body size and then ultimately converged to where all of the treatments were identical in terms of how big they were. So, hovering around the average of around 50 millimeters. So this was not at all what we expected with this study. We had expected basically these size differences to remain consistent through time to where the variability was all of these days would have looked like this first day. But when these individuals underwent metamorphosis, so if we look at this graph where we have mass at metamorphosis, so basically how much they weighed for our 3 different treatments, they were identical in how big they were, and all of our treatments had equivalent survival. So all of the survivals were just under 100% for all of our 3 treatments. And so what was going on here is something called compensatory growth, basically catch-up growth. So salamanders that hatched over those much more variable time periods were able to accelerate their growth and catch up to salamanders that had hatched on one date to result in equivalent sizes at metamorphosis and equivalent survival. And so, and they had very equivalent development rates. And so this was sort of interesting to us because it showed that despite hatching at later dates, there was no apparent cost to the larvae in terms of how big they were when they underwent metamorphosis. And this is an important metric to understand because oftentimes how big they are at metamorphosis is important for predicting how, how long they're likely to live as an adult, how many eggs they're going to lay, as well as how quickly they're going to reproduce as an adult. And so this is a sort of good proxy for what those organisms are going to do after they leave the wetlands. And so in thinking about this in terms of climate change, this shows that these animals have some capacity to deal with natural variability in the timing of their breeding. In this case, we were looking at the degree of synchrony in hatching. So you might imagine similar sorts of patterns could potentially emerge for some species if they're shifting their average date of breeding or the first date of breeding or those sorts of things. So, so this does give me hope that even if salamanders are shifting the timing of when they breed, that they're going to be able to compensate that to some degree, but probably not for any amount of climate change or a large amount of climate change. They might not be able to keep up with the rate of change, and then we might see some of the more negative impacts that might occur. So I'm going to bring back sort of the unknowns that I initially typed up at the beginning, sort of in my conclusion slide here. And so I mentioned that wanting to know where and what type of shift and who's shifting are happening, as well as what happens to those organisms later on in life in terms of what happens to populations, communities, and ecosystems. And so some of the work that I think I showed here, especially this bit at the end, that there are some consequences for populations, but they aren't necessarily all bad. But I would say we're very much in our infancy of understanding how things like phenological shifts will alter amphibian populations and communities. And so additionally, some of the data that I showed on who's shifting and how much, those studies that I showed were data from 4 years, for the one study of ringed and spotted salamanders, where we tracked their breeding migrations for 4 years. That was a Herculean effort to get that data, and it was still only 4 years. And so it just wasn't enough to say, that these shifts are occurring in terms of the timing of breeding. And so we're still trying to work out generating long enough data sets to be able to say, who and how much these organisms are shifting. And so some of the things that I think are going to help with this, that I think, that I'm working on as well as others are working on in terms of documenting these shifts, is refining some of these automated technologies to be able to generate better data sets for longer, as well as using these community science data sets and projects. So things like iNaturalist, or there's another program called the USA National Phenology Network, to be able to build these large community data sets that then will tell us a lot more, again, than individual researchers could do by themselves. And continuing some of my work, I'm continuing to do some field and experimental work, trying to understand how do these shifts ultimately affect amphibian populations. And so things like sex ratios or population abundances or some things like that to help understand what it means for these organisms. So with that, that is all that I have to convey today. So I'm happy. I saw there's several questions in the chat, so I can pull that up and start going through those. All right.
Yeah, if you want, I can go ahead and read out the questions for you so you don't have,
Yep.
so you can focus on one thing at a time there. I know how that can be. Okay, so Jeff would like to know how much / what are the leading causes of salamander mortality associated with breeding migration? And he mentions that they had a mass mortality event on a parking lot surface that was treated with road salt in a local forest preserve.
So there's a variety of things that can go on as they undergo these migrations, both in terms of human impacts as well as natural impacts. And so in terms of mortality, some of you or many of you may have seen as you drive past wetlands that are next to roads, as salamanders or frogs are undergoing these migrations, a lot of them do unfortunately get hit by cars. And so a lot of communities that have natural wetlands nearby, have salamander brigades of folks out there with buckets, picking up the salamanders and helping them cross the road kind of thing to try and limit some of that car-induced mortality of amphibians. But I think Jeff brings up a really good point about things like road salt, having a big impact on amphibians, especially around here, and as well as further north, that, the timing of when amphibians are out moving around and they would be exposed to these things could be a big problem, especially if we have late snowfall events, more towards March, that, that excess salt, knowing how much they spray on the roads, really could have a big impact if all of that drains right into wetlands right when all of these adults are in these ponds. And so that could be potentially a big problem. Salt pollution in general is sort of a big topic right now for a lot of folks, knowing that a lot of areas are increasing in how salty they are for freshwater habitats, both in terms of fish or other sorts of organisms that are occurring in these habitats. So definitely that. And then a brief part on the natural mortality causes. So raccoons and other organisms are also really good at timing when they're out hanging out around wetlands. And so raccoons and other things will be out there trying to catch and eat salamanders as they're undergoing these migrations. So there is some natural mortality as well.
Okay. Don asks, is the compensatory growth based on food availability?
Definitely is a factor that plays into this. So if there's a really low food availability in there, there's not enough food for them to actually accelerate their growth. So we set these up for these mesocosms to where they basically started out having a self-sustaining amount of zooplankton, which is the primary food source for these organisms. And so sort of after we set it up, we quantified the amount of zooplankton that was in there. And I wouldn't say that it ever got to depleted levels, but it definitely was lower. So we saw a compensatory growth despite some food limitation, though, definitely if they got down to really low food availability, there probably just wouldn't be enough for them to be able to catch up without having some sort of cost in terms of they might be smaller at size at metamorphosis if they weren't able to catch up fully. So definitely a good question.
Lisa asks, does depth of the water have any impact on the speed of maturity?
Depth of the water directly, probably not, but as depth of the water would directly impact water temperature, then that definitely could have an impact. So development rates generally are higher under warmer water temperatures. And so if it's a really shallow wetland, then probably those amphibians are going to undergo or complete their life cycle, at least in the aquatic phase, much more quickly. They might be able to undergo metamorphosis faster. And so that could have an impact on how quickly they get out of the wetland. In terms of getting to an actual size at maturation, it gets a bit more tricky just because it takes them sometimes several years to grow in the terrestrial environment before they can become sexually mature. And so it's sort of the warmer water / depth of the water plays a role, but then there's still some unknowns that could happen that could speed up or slow that down once they reach land again.
Jeff asks, are there any noteworthy phenological shifts in predators or prey that might affect amphibians?
So in terms of amphibians themselves, some of the work that I've done and I'm interested in, as well as other folks are interested in, is interactions between amphibians that could function as predators or prey on each other. And so, for example, if salamanders are shifting their phenology relative to frogs, which they might potentially be eating at the tadpole stage, then there's definitely some folks that have noticed sort of shifts in that direction where sometimes salamanders will breed earlier than frogs, and then that results in a higher mortality of the frog species. And so that's sort of related to a general concept for a lot of phenology work in this idea of something called a match mismatch, where some organism has a required resource that they need to be synced up with in terms of it being around when you're around. So for example, plants and pollinators, they need the flowers to be open when the pollinators are flying around. So there isn't something necessarily equivalent where there's this really strong obligate relationship that I'm aware of, but definitely the shifting of these, the timing of breeding can impact both competition and predator-prey interactions.
Okay, let's see. And I feel like you kind of already hit this, that Don sent, does the phenology of predators have an impact on survival? I guess that's a little bit different, but.
Yeah. And the only thing I'll say in addition to that is, so the phenology of other organisms that would eat these amphibians, so things like dragonflies, the dragonfly larvae are often big time predators of amphibians. And so they're undergoing similar sorts of shifts of, the timing that dragonflies will undergo metamorphosis out of wetlands or when they breed are also shifting. And so there's a lot of moving pieces of all of the different organisms that live in these wetlands in terms of the timing of when they're doing things that could, sort of rearrange how that community is structured.
Okay. And Bob asks, is the breeding schedule affected by moonlight or artificial light? So, all of our light pollution, I imagine.
Yeah, no, that's a good question. I'm not aware of anything that has demonstrated that that they'll migrate more under dark skies versus full moon or anything like that. But I think the artificial light thing, or both of these in general, is very underexplored in terms of their impacts. So knowing exactly how if a day length is a typical cue that these organisms might also use in addition to temperature and rainfall, since day length would be a much more consistent and reliable indicator of the season as opposed to things like temperature, how we're affecting that with things like artificial light. I think we don't have a good understanding of what those impacts are. And we know that artificial light has an impact on plant phenology. So artificial lights can impact the timing of how long trees hang onto their leaves, as well as, when things, leaves emerge. And so, there's definitely the potential that those sorts of things could have an impact.
Maxwell asks, what do you feed the captive salamanders?
Yeah, so thankfully for those sorts of experiments that I mentioned for that study, we really don't have to feed them a whole lot in terms of active feeding. So when we've developed those pond ecosystems, we put in a variety of leaves, dry leaves, and sort of pond inoculum, we call it. Basically, we go out and take a water sample from a natural wetland and pour that water in to then inoculate it with algae and zooplankton and a variety of other critters. And then the tanks are open generally so that mosquitoes or other organisms can lay their, other insects can lay their eggs and developing larvae in those, and those are food for salamanders as well. And so basically it becomes sort of a self-sustaining aquarium, for lack of a better word. So we don't have to feed them a whole lot beyond what we initially start the tank, the initial conditions of the tank. So they're really nice because they're low maintenance.
Kind of my own question to follow on with that. Do you have a problem with predators finding and kind of scavenging from your mesocosms?
Generally, no. I've never really had problems with that. The tanks that I've used are, I don't know, 2.5 feet off the ground. And so they would have to be pretty substantial to be able to crawl in there. And it seems like things like wading birds, like herons and whatnot, just they don't recognize that as a place that would be an easy buffet land for them if they could figure it out. So, I generally haven't had issues like that. So thankfully, fingers crossed.
Yeah. Hopefully it stays that way.
Yep.
And then another question, did you account for cannibalism in your study of the larval marbled salamanders in the Mesocosm?
Yeah, so that's a good question. So this experiment that we set up was really geared towards trying to understand whether or not shifts in the timing of breeding would induce cannibalism. And we really didn't see it because basically, if I go back to the graph of survival, go back a little bit. There was this little bit of a dip in survival in this medium synchrony treatment, but otherwise, survival of the 36 individuals that we put in these tanks, we reliably got, between 33 and 36 out of them. So, we, there might have been some cannibalism, and we saw some missing tail tips and some of that sort of stuff that they tried to eat each other. But, in terms of overall individuals getting consumed by others, it probably was pretty low, which we were really shocked by. We thought, these salamanders are known to be really voracious, and we definitely thought they were going to eat each other. And so, what I think was going on, and this is actually going to answer Lisa's question next. So these experiments, so the marbled salamanders breed in September and October, and so this experiment was set up in October or so, and that's usually by the time most dragonflies have stopped flying around and laying their eggs, and so we don't have to leave them covered over the winter just because there's no major insects at least flying around. In the spring, there's maybe a couple week period where the dragonflies had started to come out and lay their eggs again. But at that point, if they're laying their eggs in these tanks, then that's just extra food for the salamanders, because the salamanders are big enough at that point, they would eat the developing dragonfly larvae. So for this experiment, we did not have to worry about that. But had we done this over the summer, we definitely would have had screens on top to keep out unwanted things colonizing them like that. But back to the other question about cannibalism. So what I think could have been going on is basically that because this was done over the winter, for the most part for this experiment, that water temperatures might have been low enough that their metabolism was low enough that they just weren't actively trying to seek out and catch other salamanders that might be able to swim away fast enough, just because they were, it was a cold water temperature and they just weren't as active around there. So that could be one reason why we didn't see a lot of cannibalism relative to other studies, but it's a good question.
Hey, well, that's all the questions I see in here. Oh, if anybody else has another question, we'll give another minute or two, and you can also unmute and ask it yourself if you'd like. In the meantime, I'll just say I want to thank Tom for the great presentation, and thank everyone else for joining us tonight. Before you go, I want to mention that our next Winter Speaker Series event is tomorrow night at 7 p.m. Megan Stropus will be joining us from Joliet Junior College to talk about her work comparing the colonization of aquatic macroinvertebrates in different types of habitats. And you can learn more about this event tomorrow night and other future activities on our webpage or our Facebook page. So you can just look up RiverWatch on Illinois RiverWatch on Facebook, and it's all there.
And I would say that if you do have further questions and we're too shy to ask, feel free to email me anything too. My email address is listed there. So, feel free to email me if you have further questions about this.
All right. Well, great. Thank you again. And I hope you all have a good night.